So you've covered mitosis and you want meiosis. Well, here we go with the lecture on meiosis and how it affects heredity. So during this lecture, please be able to define meiosis, haploid, diploid, homozygous, heterozygous, crossing over, law of segregation, law of dominance, law of independent assortment. Be able to list and define the phases of meiosis. They're very similar to mitosis, and we're going to be talking mostly about the exceptions during this lecture. Differentiate between meiosis 1 and meiosis 2, and relate meiosis to heredity. We're going to be using a term here called homologous chromosomes. So what are these homologues, as they're called? Well, as we mentioned earlier, you get one set of chromosomes from your father and one from your mother. And this means that for every chromosome, like chromosome 1, chromosome 2, chromosome 20, you have 2 copies. Now, those are called homologous chromosomes when you have 2 copies of one chromosome. Alleles are essential in this because, as we mentioned earlier, an allele is a variant of a gene. And you may have 2 copies of a gene that are the same. If they are the same, and that means they are the same alleles. And if you have 2 copies of the same alleles, then you are what is called homozygous for a character. So for say blood type, if you have type A and you have 2 copies of the type A allele, you are homozygous for type A. An organism with 2 different alleles for a character is heterozygous for that character. So if you have one allele for A and one allele for B with your blood types, you have type AB blood and you are heterozygous. Now, when it comes to which alleles you have, this is the genotype that you have. So these alleles are in regard to genotypes. We can see here, if we were looking at some pea plants that have either a mutation in the gibberellic acid or not a mutation in gibberellic acid, then we can have the T for tall allele for no mutant, and T, lowercase t, for dwarf allele, which means they have a mutation in the gibberellic acid pathway. So the genotype here is going to be big T, little t, and that will be a heterozygous genotype. With meiosis, we are moving from a diploid cell to haploid cells. So let's look at interphase first off. We're going to start with a pair of homologous chromosomes in your diploid parent cell during phase G1. And then during S phase, that's going to be replicated. So now you have sister chromatids for each one of those chromosomes. G2 phase progresses, the cell grows a little more, and then the cell begins meiosis instead of mitosis. The first thing that happens is there will be a separation of the homologous chromosomes during meiosis 1. And that means the cell goes from diploid to haploid, because instead of having 2 copies of the chromosome, they now only have 1 copy. That copy is made up of sister chromatids still, but now this is a haploid organism. In meiosis 2, those sister chromatids will separate. And honestly, if you know this slide, you're going to go very far in questions about meiosis. But we're going to look into the phases of meiosis and what makes them special. This is meiosis writ large. So what I want you to see here is kind of an overview. In meiosis 1, the homologous chromosomes separate. In meiosis 2, which is mislabeled here, the sister chromatids will separate. So in meiosis 1, we have prophase, metaphase, anaphase, and telophase. There is a prometaphase in meiosis 1. It's just not shown here. In meiosis 2, we have prophase, metaphase, anaphase, and telophase. There is a prometaphase. It's just not shown here. And for each of these, we label them 1 or 2. So here's a good clue. If I'm talking about, is this a phase of meiosis or mitosis? If it has a number after it, it's meiosis. If it doesn't have a number after it, it's mitosis. During prophase 1, you can see the homologous chromosomes pairing up, and you can see something formed called chiasmata. We're going to get into that a little more often later. Metaphase 1, you can see that the chromosomes are lining up, but they are pairing up with their homologue on the metaphase plate. Anaphase 1, those sister chromatids are attached, the homologous chromosomes separate. Prophase 2 through telophase, you have technically a haploid cell. In prophase 2, you're going to have those chromosomes they're not forming chiasmata because there is no homologue. Get to that more later. Metaphase 2, you have them lining up on the metaphase plate. Anaphase 2, the sister chromatids separate. So that's the overview. Let's get into the details. With meiosis 1, we have something called prophase. And during prophase 1, there is crossing over. What this means is that the sister chromatids are still attached to one another, but they are going to link with their homologues and exchange genetic information in a process called recombination, where you essentially break off an arm and separate it with the equal and same arm of the homologue. This crossing over creates new combinations of alleles and occurs during prophase 1 of meiosis. During metaphase 1, we have something called independent assortment, where we're going to have the homologous chromosomes line up on the metaphase plate. So during anaphase 1, these homologous chromosomes will separate. So let's look over here. Remember, meiosis 1 makes haploid cells. You're inheriting 23 chromosomes from each one of your parents. Each non-reproductive cell in your body will have 46 chromosomes, but you're going to make that down to 23 chromosomes in the gametes by separating the homologues in meiosis 1. Look here at metaphase 1. What we see is there's a metaphase plate, just like metaphase of mitosis, but here the homologues are lining up, and that's what's special about metaphase 1. You get independent assortment of the homologues along the metaphase plate. During anaphase 1, these homologues are going to separate, and as the homologues separate, you are going to form two new haploid cells after telophase 1 and cytokinesis. Let's look at meiosis 2. In meiosis 2, we start with a haploid cell in prophase 2. During metaphase 2, the chromosomes line up along the metaphase plate. During anaphase 2, the chromatids separate just like they would in mitosis. During telophase 2 and cytokinesis, it's very similar to what goes on during mitosis. Meiosis 2 is essentially the mitosis of a haploid cell into the individual gametes. There are some laws of heredity that come into play when we look at meiosis. The first one is the law of segregation, which is when gametes are formed during meiosis, homologous chromosomes separate from one another and go into separate gametes, which means the alleles separate. Each gamete can only receive one allele for each gene. This is how this works. Let's say you had that pea plant that was heterozygous for the tall or short dwarf alleles. You have a diploid cell to begin with, and it's going to have your sister chromatids on your homologues, and those homologues will pair with each other, and during metaphase 1, the homologues will separate. After telophase and cytokinesis, you now have two separate cells. Each one of these cells is haploid, and each one of these cells has only the same alleles. So what we have is the lowercase t's are all in one spot, and the uppercase t's are all in the other spot. They are segregated into separate cells. And during meiosis 2, we're going to make 4 haploid cells, and that will further separate these different alleles. The law of dominance. If you have 2 alleles, but only one trait is observed, that gives the phenotype. This is the dominant allele. The other allele is a recessive allele. We remember this with the O blood type. The O blood type was recessive to either A or O. If you have the A allele and the O allele, you have type A blood. If you have the B allele and the O allele, you have type B blood. The description of what blood type you have is your phenotype. The genetic makeup, having both alleles, is your genotype. Can organisms have the same phenotype but different genotypes? Well, let's take a look. We have here 4 different pea plants. These pea plants have different alleles for either the purple allele or the white allele. Uppercase P is purple, lowercase p is white. And what we see is having both copies of the purple allele gives you a purple phenotype. Having one copy of the purple allele and one copy of the white allele makes you heterozygous, but gives the purple phenotype. So yes, you can have different genotypes, but the same phenotype. And what we also see is that there is roughly a 3-to-1 ratio in the offspring when it comes to the different phenotypes. You have 3 purple to every 1 white. And the genotypes is 1 to 2-to-1. Last, the law of independent assortment. Each pair of alleles will segregate independently of other alleles. So this means there are different combinations of 2 traits. If we look back at metaphase 1, you saw that the different chromosomes didn't all line up having the father's chromosomes and the mother's chromosomes when they paired with their homologues. And when they segregated during metaphase 1, they segregated independently of one another. And generally speaking, if we have alleles that are on different chromosomes, they are going to segregate independently, leading to the law of independent assortment. This happens during meiosis 1, and this assures that unrelated traits stay unrelated. So, say hair color and a prevalence towards diabetes will stay unrelated if they are on different chromosomes and segregating independently. Here we have another example of maternal sets of chromosomes and paternal sets of chromosomes, where you have 4 different possibilities of how they can sort into different cells in metaphase 2, and this is equally possible depending on how they arrange in metaphase 1.